Hello everyone, welcome back to my channel Botany for NEET. Let us discuss a question asked in NEET 2023 paper. Let's read the question. In tissue culture experiments, leaf mesophyll cells are put in a culture medium to form callus. This phenomenon may be called as dedifferentiation, development, senescence, differentiation. Before solving the questions, let's understand the question. Tissue culture experiments. So basically here, usually we take a Petri plate and into the onto the Petri plate we add medium, which is supplied with rich source of nutrients. So basically here, any tissue of the plant is taken, allow proper sufficient temperature and nutrients are provided such that when the tissue is grown on the Petri plate containing the medium, then it develops into a complete plant. Now, in this tissue culture experiments, leaf mesophyll cells were put in the culture medium. So, culture medium is nothing but a Petri plate. You take a Petri plate, onto the Petri plate, you add medium. Okay, so basically, media is nothing but substance which is rich source of nutrient to form callus. So, what is the function of mesophyll cells? So, mesophyll cells they play a very important role in the process of photosynthesis and also in gaseous exchange. These mesophyll cells are of two types: palisade mesophyll cells and spongy mesophyll cells. Now, what is callus? Callus is a mass or a group of undifferentiated cells. That means these cells they continuously divide. They do not have any specific function. They are not specialized to perform any specific function. So they continuously divide. So in tissue culture experiments, when a leaf mesophyll cell is put in a culture medium, it forms callus. So this phenomenon is called what? So let's consider an example of root tip. We all know that the root tip contains meristematic cells. So these meristematic cells continuously divide. They are not specialized to perform any specific function. So these meristematic cells later they got specialized to form root hair cells. What's the function of the root hairs? They basically help in increased absorption of water. So here the root hair cells they now perform a specific function and they have lost their ability to divide. So this process where meristematic cells, the cells that continuously divide, gain a specific function and lost their ability to divide, that process is known as differentiation. The process where meristematic cells get specialized to perform a specific function and lose their ability to divide is called differentiation. Now, these root hair cells, when they again regain the capacity to divide and they are not specialized to perform any specific function, so they are meristematic. Again, that means they can continuously divide, they have the ability to divide. So this process where differentiated cells, the root hair cells, the differentiated cells, again they have regained the ability to divide, that process is known as de-differentiation. So differentiation is a process where meristematic cells have lost the ability to divide to get specialized to perform specific function, while de-differentiation is a process where differentiated cells have regained their capability or capacity to divide and are not specialized to perform any specific function. So now let's read the question. Here, when tissue culture experiments were conducted, the mesophyll cells, where the mesophyll cells are differentiated cells, when they are put in the culture medium, they form callus. Callus are group of undifferentiated cells. They do not have any specific function. So they became again meristematic. So, what is the phenomenon involved here? De-differentiation. So, the process by which the completely differentiated basophil cells again they have regained the capability to divide to form callus. That process is called de-differentiation. Okay. Differentiation we have already discussed. Senescence means to attain old age. That means when the plants start develop growing big and the leaves start falling. So senescence is aging process. Okay. So basically for the question, in the tissue culture experiments, leaf mesophil cells are put in a culture medium to form callus. This phenomenon may be called as de-differentiation. So the correct option for the question is option 1. I hope you understood the solution for, for this question. If there are any doubts, please feel free to comment down below. If you like the video, please share it with your friends. And for more such content, subscribe my channel. Thank you. Have a good day.